All right, welcome back. I figured because of my technical difficulties, I should just go ahead and finish off the lecture right here. And also, this is going to be a SARS-CoV-2 related COVID point. This is how to look at these trees, and we'll just finish up talking about sequence alignment trees. When you get all these DNA sequences, you can put them together in a tree, right? And so what can you do with that tree? It's surprising that you can do pattern recognition of some sort with the tree. So if you just look at a tree, you can tell if it's plausible or not. This is actually an important point because with SARS-CoV-2, you ended up with this case right here. There was, this is very early on. If you look at the date, this was April 23, 2020. So a long time ago now. We were just figuring out how SARS-CoV-2 was spreading and mutating and things like that. And what we did is that we took the different RNA sequences. Technically, it's an RNA genome, but works exactly the same as DNA. And we can tell where the proteins are in that. We can tell what the mutations are. And we can see where SARS-CoV-2 picks up different mutations. Now, the important thing about SARS-CoV-2, it has a huge pool of possible mutations. That means that we do get mutants. But it is not a particularly fast mutating virus. Coronaviruses are not the fastest. There are other viruses that mutate faster. So it could be worse. The reason is because of the way their polymerase works. The way that they work, they just don't make as many errors as other viruses. That means that when you compare, especially this early on in the epidemic, you would have a handful of base pair changes, maybe not even changes in the protein sequence. And what you would do is you would collect these 5 different sequences, for example, and you would see the blue mutation in all of them. You would see the red mutation in D and E. You would see the green mutation in A and B. From this information, you can infer what a tree would look like, that you have C closer to the root of the tree. You have, the blue mutation was closer at the root of the tree, and it shows up everywhere because it happened before any of these diversified. And you can put together a sequence, and you can put together a tree. Now, the thing about the trees is you can look at their shape, and even not knowing anything about the actual mathematics of how this was done, you can at least look at the tree and say, does that tree support what the person was saying? The reason why this is important is because we had this case right here, which is shown by Twitter. There are many things that are terribly wrong about this paper. There's a paper that came out in PNAS, which is a prestigious journal, but it has an issue. It has a fast track that, like the special elite scientists, members of the academy, can get their papers published a little more easily than other ones, maybe without the proper peer review. And it looks like that happened in this case. A bunch of scientists published this tree, and you see that they said, and here is another scientist doing peer review in real time, where they're taking the tree and they're saying, there's a problem with this tree. This doesn't look like it should. So there are 2 things that this scientist pointed out. For example, they said that they were comparing the bat sequence to the other ones. If you look at this tree, literally published in the paper, you'll see the bat sequence is on this really weird little branch. If you walked around campus under a tree that had a branch like that, you would be worried that that tree is unstable and that branch will fall off and hit you on the head. In the same way, this tree looks unstable because the bat is just too far away, it's sticking out too much relative to everything else. So I can say that's fishy, and I can look at another scientist, and the scientist says, I know this stuff better, and it's really fishy. I can agree with the scientist who says it's fishy. The other thing that they said is the main thing of the paper is that they said they found 3 distinct clusters. In fact, that's why it made the news. They said, oh, SARS-CoV-2 already is forming 3 distinct clusters that act differently. It's already mutating, and maybe it'll become a zombie-eating virus next or something like that. And that's literally how this was reported. The 3 distinct clusters, they showed this figure, and they said that we see 3 clusters there. And I don't know. I don't see 3 clusters. Maybe if you squint, you kind of see the A, B, and C being a little bit different. But just like you would say that's 1 tree, not 3, you can look at the shape of that and say, hey, I think the Twitter guy's right. You don't have to know everything to be able to draw some conclusion about what's reasonable. And it's not reasonable to say that this tree has 3 distinct clusters. So use your pattern recognition. Look at the tree. Make a guess. Be uncertain about your guess, but go ahead and make it. And that's what I'm asking you to do in the lab as well. So, there's one more way you can look at aligned sequences called a sequence logo. You can, when you align the sequences, like remember the cytochrome C had that stretch from 70 to 80 that was all the same. We would call that the consensus sequence. All the different sequences had a consensus that 70 through 80 would be the sequence that we showed you. You can do that for other sequences as well. At the top, it's done for a short peptide. At the bottom, it's done for a longer sequence, actually 8 residues on the top and 13 residues on the bottom. They aligned a bunch of sequences, and they can represent exactly where the variation is they got in brackets at the top. And you can see for the bottom one, that's just weird. It's just ridiculous. Nobody can read this. But this exact same information is being put in graphical form, and you can read that. So I want to say that each of these is a binding sequence. That means it's going to bind something of complementary charge. Negative things will bind positive things. You know, it's that kind of thing. So look at the top sequence and tell me what charge you think will bind to this. When you look at that, you see that there is the lysine, K, made helpfully blue for you and made really big because it's a consensus among all the sequences they compared. You can see right next to it, you can have a threonine or a serine, less of a consensus, but shown as a sequence logos way, there's still like half of the sequences have threonine, half of them have serine. So it's a very intuitive way to show. You just make the consensus big. And so it turns out that this is actually an ATP binding section because ATP has those phosphates that are negatively charged. And so you have the positive lysine to bind the negative phosphates. On the other hand, the other one has the opposite situation where you have 4 red residues which are acidic and negatively charged. It turns out that this is a calcium binding site. And all the sequences, they can have lots of variation in other places, but almost all the sequences have D's in the middle, and all of the sequences have a D in position 1 and an E in position 12, except for a few of them that have, see that little squished D down there? It looks like some of them can get by with a shorter version of the same amino acid, but they need the negative charge. And it turns out that this is a calcium binding sequence. Calcium is positively charged, so it makes perfect sense to have 4 negatively charged residues. And you can take this consensus sequence. If you have a sequence you don't know what it does, but it matches the consensus, you can say, hey, I think I have a hypothesis here, guys. I think I know what this might do. And then you do an experiment to check it out. This is what cytochrome C's logo representation looks like, and the only thing is it's a mess, even for a small protein. But if we zoom in on that sequence in the middle, you see how you have the sequence around 74. That's the exact same thing that we showed you, that has all of the same stuff over and over again. And in fact, I showed that to you again. The invariant yellow residues are, they show up as big single letters or almost single letters in this logo's representation. Now this logo was actually made by a different set of sequences. That shows you again just how robust our finding is, that this sequence here, 70 to 80, is always the same in cytochrome C. There's one final thing to talk about. When we talk about the different cytochrome Cs in different species, those are orthologs. Orthologs are similar sequences in different species. If you talk about, there are also homologs. Any 2 sequences that are similar are, we call them homologous, and we call them homologs. So all orthologs are also homologs. But there's also a different thing where you can have similar sequences in the same genome. You can have one that's a slightly different sequence. You call the first one alpha, the second one beta, maybe. Those are called paralogs for being homologs that are next to each other, para. And so there's a big difference between orthologs in different species and paralogs, same species but different genes, different similar genes. And that's really interesting because you can see, for example, for globin genes, you can see that most mammalian and frogs, chickens, and mouse all have 2 copies of a globin gene. We call one alpha and one beta. So those are orthologs, and we're comparing the alphas among frog, chicken, and mouse. Those are orthologs. but the mouse alpha and beta chain are paralogs with each other. They're all homologs. And this tells us something about how the proteins were made. It looks like there was some ancestral mutation that made an alpha chain and a beta chain. And then all of these other animals inherited those two chains. And you can talk about, are the beta chains more similar, or are the paralogs more similar? Is the beta more similar to alpha in mouse, or is it more similar to chicken in beta? And that's an interesting question. There's lots of stuff going on. And actually, there's projects that we've been involved with that look at what are the function of paralogs. Why do you keep two copies of a globin gene around? It turns out they do two different things, but they can have very similar structures. They at least have very similar primary structure, because we talked about, that's what we're talking about today. We'll talk about the other levels of structure later. And so the amazing thing is that you can find homologs of all your genes down to bacteria. And that's why studying bacteria can actually give you a biochemical conclusion that then you can apply to humans. We're very different organisms, but our genes are surprisingly similar. And there's all sorts of evidence for this. You can look into this yourself. And you can even plot a chain of transmission. You can even plot a mechanism of evolution in certain cases for how a gene was duplicated and then diversified. The two paralogs got different jobs. It's just, now that's more for genetics and molecular biology, but we do talk about it a little bit in biochemistry. In fact, we talk about it at the end of chapter 3. And so we are done. I showed this before, so please be ready to talk about the reading guide as assigned on Friday. And I will answer all your test questions there for everyone to hear. Then we'll be ready for the test on Monday. All right. I will see you either in the lab or in lecture. Thanks for listening.